I'm Dan Garry from the University of Minnesota, and I'm here with Laura Zelarayán. Laura?
Yeah, I'm Laura Zelarayán from the University of Gießen in Germany. Yeah.
And Laura just gave us a terrific talk
Thank you very
focused
much.
on KLF15 and using emerging technologies. Laura, would you mind just telling us briefly about your project and your presentation?
Of course. Yeah, so our project is about to find a target in order to revert heart failure progressions and to, let's say, diminish a really medical need, which is the cure of heart failure. So we have studied models in a cross-species study, studied mouse and human models, and determined the importance of one transcription factor, which is actually known to play a role in homeostatic transcription in the heart, which is a CRISPR factor 15, which has transcriptional repressors and activation roles. And we have found that it's a very crucial factor whose activity is reduced during the course of heart failure. And what we did is we developed a CRISPR activation approach to target the heart in order to recover the transcriptional activation of this CRISPR factor 15 in order to resolve heart failure progression.
What can you tell us about KLF15? Where is it expressed? How broadly is it expressed within the heart? And what do we know about its transcriptional program?
So CRISPR factor 15, it was actually discovered in the kidney, and that's why it was called KLF15. But it's also expressed in the heart and is expressed in cardiomyocyte and in fibroblasts with different transcriptional programs. It's known to play an antifibrotic role in the fibroblasts, and we discovered that it's also playing an antifibrotic role while inducing a specific transcriptional network in the cardiomyocyte. So basically, KLF15 is very important for a network of homeostatic program for metabolism, so fatty acid metabolism, but it's also important to prevent the activation. So it's a repressor of pathological hypertrophic program. So the valence is very important when the factor is present. So what we do with our approach is to bring back this factor in order to push homeostatic programs and to revert the activation of pathological hypertrophic program. And this is a huge network that is all done by these factors.
Your results were truly remarkable, showing that by overexpressing KLF15 that you could improve cardiac function and decrease cardiac dimensions following TAC. Can you give us some sense for the time course that took to see those types of changes?
So the time course is something which is very crucial, and so far we are depending on adeno-associated viruses delivery, which also is accompanied with a transitional, a little bit of time that you need a transduction and effect in a specific cell, in this case, the cardiomyocyte. So in the mouse model, we are talking about a couple of weeks. So when we apply our AAVs, the older the animals are, the longer you need of the time to have a transduction and an effect in your specific cells. In vitro, when we use human cells, of course, it's a little bit faster. So I believe that we will have to wait a couple of weeks with this approach that we have at the hands and at the moment, which are the AAVs. And as we heard also in the same session, there are other approaches that are in development, like lipid nanoparticles or other kind of particles that can be used that I believe that we will succeed in being faster in delivering our approach once it is discovered.
Now, you used aortic constriction, aortic banding to induce heart failure. Have you looked at other models, or do you plan to look at other models of heart failure? So is this more broadly applicable to other types of heart failure?
This is an excellent question. So what we are trying to find is the concept of precision medicine. So for every disease, we would probably have a specific factor that we have to target. Our single cell analysis that had show us that this factor is important is in the transaortic constriction model, so in hypertrophic remodeling, but we also tried in ischemic remodeling. And in that case, we didn't see a major change, but of course we had to continue with other kind of cardiomyopathy, like diabetic cardiomyopathies, and to see whether there we have also a stent abscess. But for ischemic cardiomyopathy was not the same result as we have seen in hypertrophic cardiomyopathy.
And do you plan to translate your studies from small animals, rodents, to larger animals?
Yeah, we do. We are actually doing with a big group of people because we really need a lot of expertise. Many of our colleagues are based in Germany, and which are the experts of a large animal model. We are trying the pig model at the moment, and we are introducing our CRISPR approach in a non-transgenic manner, and also inducing hypertrophic remodeling and see whether this large animal model we can go further also to a clinical trials. And this is something which is ongoing. Yes.
For patients that have heart failure, uh, maybe you can just try to reduce your findings to how does your basic science findings, what does that mean for them? And what is your particular approach? What would this suggest for treatment of heart failure?
Yeah, by coming to the patient, I will ask my question, and I work in team with cardiologists because I'm a purely basic scientist. I'm biochemistry, so I'm trying to understand every molecule, how they change the course of a cell fate. And I guess for the patient, this is not that important. For the patient, it's important to know that it's safe. And this is where we are working very hard. And also, as I said, teaming up with cardiologists that tell us what is the need, what are expecting the patients to give for curing the heart failure. Heart failure is a big umbrella. So we really have a lot of, many of them are also genetic cores that we probably don't know. But having one core for a more broad approach of heart failure, I believe this is what the patients would like to have in order to not only to prolong survival. I think what we want to do is to help the patient to have a longer life with good quality of life. This is what we are trying to develop. Yeah.
And maybe you could just kind of emphasize what's next? Where do you go from here? Not only with regards to your scientific efforts focused on heart failure, but also using this CRISPR-based approach.
So actually it opened a lot of doors for us in the lab. So we came across this for a technical reason, and now we see how many potential avenues are open for this approach. So we are, of course, doing some screening to know how many other diseases can be cured, regenerative approaches for ischemic disease. So this is what we are trying now to apply what is very well established, as I said before, in different disease conditions, and to see how we can even exploit more this multiplexes approach. You can also target many targets at the same time. And this is where we go now. We want to engineer networks or gene networks in order to imitate what is happening in the body. And this is where we are concentrating now our let's say, basic fundamental approaches at the moment, and without losing the focus of translating this in a later time point into a disease therapy. Yeah.
Heart failure, as you know, it's devastating, and the morbidity and mortality is tremendous. 50% of people following diagnosis within 5 years are not alive. And so these kinds of therapeutic approaches and these kinds of basic science approaches for which therapies can be built upon are absolutely essential. Thank you for all your work, and keep up the great work.
Yeah, thank you for your interest in, and yeah, I'm happy to contribute with our basic science.
Okay, thank you.